Hi, everyone, and welcome to today's lecture on DNA packaging and transcription. So before we get into the details of transcription, which is gene expression, I want to talk a little bit about the packaging of DNA because keep in mind, before you can express those genes, the genes and the DNA have been tightly coiled and compressed to fit inside of the cell and if it's bacteria and inside of a nucleus, if it's a eukaryotic cell like our own. And so packaging of DNA is very important in this context. Now, first, we'll take a look at bacteria. And when we talk about bacterial packaging of DNA, what's necessary first is called supercoiling. Okay, supercoiling is necessary to fit the DNA into cells, and you see all of this ultra coiling here of DNA. Now, the way that that happens is through an enzymes, which are topoisomerase 2 enzymes called gyrase. And gyrase is something that is in bacteria but not in humans, and that becomes very important in the medical field because it's something that we can end up targeting when we're trying to kill bacteria too. We'll talk about that a little bit more in a moment. So now what gyrase does is it twists the double helix DNA into these giant twisting loops that extend from a center scaffold that you see right here. Now, the fact that gyrase is needed in bacteria to coil and then also in a moment you'll see to uncoil DNA, that's important because we can then target gyrases. We use gyrase inhibitors and antibiotics that target gyrase because that gives you what's called high selective toxicity, meaning that you can hurt or kill the bacterial pathogen without hurting or harming the human because the human does not have that enzyme. We don't need that enzyme. So you could then target it. Now, with eukaryotes, the packaging of DNA ends up slightly different because now you're coiling up this DNA tightly to fit inside of a nucleus. So when we talk about eukaryotes, the first term I want to mention is the nucleosome. And you can see in this figure here, the nucleosomes are lined up over here. What nucleosomes are is DNA wrapped two times around a core unit of 8 histone proteins. So those colorful little balls that you see in the center here, those are histone proteins. It's an octamer for each nucleosome. So each nucleosome will have DNA wrapped two times around a core that has two of each of the H2A, H2B, H3, and H4 histones. And it's basically approximately 147 base pairs of DNA wrapped around that octamer. And then what you see is you then have a whole bunch of nucleosomes that are linked together. And so the entire length of all of the nucleosomes, lined up, coiling the DNA, folded up, that makes up what is called chromatin. And chromatin is critical for fitting all of that DNA into a little tiny nucleus. And there are two terms that I want you to know when it comes to chromatin. There's heterochromatin and there's euchromatin. So for heterochromatin, and I want you to circle star highlight these terms, heterochromatin I want you to think of as repressed DNA. In this chromatin, the chromatin is highly compacted, and so the genes are not accessible to transcription factors, okay? So heterochromatin is when the DNA is tightly coiled around those histone proteins, okay? So you can see here, if you see those nucleosomes tightly lined up together, the DNA tightly wrapped around the proteins, that DNA is not accessible to transcription factors and to polymerases or anything. Okay, so that's repressed. Heterochromatin is repressed DNA, so it's highly inactive. Whereas euchromatin, on the other hand, you are active. Okay, so that works if you're a gym bro. So euchromatin is active. That's when the DNA has been loosened away from the central histone proteins. Okay, so I don't have it on the figure here, but we're going to take a look at that in later slides. So euchromatin is active DNA because the DNA is loosened away from the histones. And so now each of those genes in that area are easily accessible to the transcription factors.
Cells use DNA to store information, while proteins are used to perform the activities necessary to keep the cell alive. In a process called transcription, the information stored in a DNA molecule is copied into RNA molecules, which can be used to synthesize specific proteins. Cells use three types of RNA: ribosomal RNA molecules, which are used to form ribosomes; transfer RNA molecules, which deliver amino acids to ribosomes; and messenger RNA molecules, which carry the information for making specific proteins from DNA to ribosomes. There are three stages in the process of transcription: initiation, elongation, and termination. Let's see how each stage works. During initiation, RNA polymerase binds to the DNA and recognizes a site called a promoter at the 3' end of the template strand of the target gene. When RNA polymerase finds a promoter, it breaks the hydrogen bonds holding the DNA strands together at the site of the promoter, and transcription begins. RNA polymerase does not bind to all promoters with equal affinity. This difference in promoter strength is one way that cells can control gene expression. The more strongly RNA polymerase binds to a particular promoter, the more likely that gene is to be transcribed. In general, higher levels of transcription lead to higher levels of translation, which leads to higher concentrations of that polypeptide. One strand of the DNA serves as a template strand. The RNA transcript is copied from this strand of the gene and therefore has an RNA sequence complementary to this strand. During elongation, RNA polymerase moves along the DNA, opening up a bubble in the DNA. As it moves, RNA polymerase adds bases to the 3' end of the growing RNA transcript. Elongation continues until RNA polymerase reaches a site on the DNA called a terminator. At this site, RNA polymerase and the newly synthesized RNA transcript are released from the DNA. Termination can occur through two mechanisms: self-termination and enzyme-dependent termination. In self-termination, the RNA sequence transcribed at the terminator causes the RNA to hydrogen bond with itself, forming a stem loop structure, which essentially pulls the RNA polymerase off the DNA. In enzyme-dependent termination, a termination protein binds to the terminator and pushes RNA polymerase off the DNA.
So, as I said before, anytime you hear the term gene expression, that means transcription. Okay, so that means going from DNA to RNA. Basically, you're building an RNA sequence using a DNA code. Okay, and in a minute, we're going to practice how to do that, making you in charge of being able to build RNA just by looking at a DNA code. Now, when we talk about gene expression, most genes in a cell are constantly varying expression. So sometimes certain genes will be turned on, whereas other times they'll be turned off. So we usually say activated or repressed. Because think about it, if all of the genes in a cell were active at the same time, can you imagine how much RNA would be produced? That cell would basically explode, right? So you only have genes active when they're needed. Now there are some genes that are always active. We call these constitutive or housekeeping genes. So circle those terms, constitutive and housekeeping genes. They mean the same thing. They simply mean that a gene is always active, okay? So it's always being expressed. These are basically genes and products that would need for processes that are always occurring in a cell. So think of life processes such as, genetic processes themselves, where you always need enzymes to be made for those processes. So for instance, polymerase genes, the genes that are responsible for ultimately making polymerases, those are always active because they're something that the cell will always need. Now, when we talk about DNA coding for RNA, there's actually all different types of RNA that's made. There's mRNA, rRNA, and tRNA. But the one that will ultimately encode for a protein is mRNA. So you're going to hear more about that in later slides. Now, keep in mind this process of gene expression. To express DNA, DNA is double-stranded, right? So those double strands are going to need to separate, just like we saw them do in DNA replication. Now, since DNA is double-stranded, but it's coding for RNA, which is single-stranded, you're going to end up with different types of strands. You have the antisense strand of DNA and the coding sense strand. Okay, now what you're going to notice in this process while RNA polymerase is building the RNA, these two strands of DNA have different roles because remember, since DNA is double-stranded and it's producing a single-stranded product of RNA, only one of those strands is going to be the template, okay, the template or code used to build that RNA, okay? So in the next slide, I'm going to demonstrate what this means, but for now, you can think of the coding strand or the sense strand as having the same sequence as RNA, except that, of course, DNA always has a T wherever RNA would have a U. So when you see sense strand, I want you to underline the S at the beginning of sense strand and write same. Okay, we're going to go over that again in the next slides, but I want you to start associating coding or sense with being the same code as RNA. Whereas the antisense strand of DNA is actually the strand that will be the template for building the RNA, which means that as the cell reads the antisense, it's going to pick the complements of that sequence to build the mRNA. So for antisense strand, you can make note that this will complement the RNA sequence. It will be anti it, so it won't be the same letters. It'll be the opposite or the complement. Whereas the sense strand will be the same sequence. Okay, so we're going to visualize that now in the next slides because you have to be very comfortable with making RNA simply by looking at a DNA code, the same way that a cell would. Okay, so to help you visualize the concept of the sense DNA versus the antisense DNA and how it relates to the RNA, let's think of it this way. Whenever you're given a DNA sequence, for instance, if you're doing any research and you look up DNA sequence online, you always find the sequence written out as a single strand in 5' to 3' direction. So when I say 5', that's 5' to 3' direction. And like I mentioned earlier, that's basically just a label of orientation, kind of like saying left to right, or top to bottom, or even north versus south. Okay, so 5' and 3' is just the orientation of DNA. Now, when you find this sequence, you notice this is a single strand, right? But what do you know about DNA? DNA is double-stranded. So even though you have this single strand, the DNA itself is actually 2 strands. And the second strand of DNA is always the complement of the first. Okay, and remember, for complements, we previously said you can use the memory trick at Georgian Court. The complement of A is T, the complement of G is C. Okay, now when we talk about 5', 3', those are the complements to each other. So the complement of 5' is 3', and the complement of 3' is 5'. Okay, so you can kind of think of them as opposites whenever I say complement. So if our DNA sequence is 5', C, A, C, G, G, C, 3', then the double strand of that DNA would be the complement. It would be the complement of 5', which is 3'. The complement of C is G, A is T, G, C, C, G, 5'. Because again, each of these is the complement of the one we see above. So the complement of 5' is 3', the complement of the C is G, complement of A is T, complement of C is G, and so forth. Okay, right down to the very last number, the 3' complement is 5'. So now, if we're going to build an RNA strand from this, what did we say about any kind of polymerase? What direction do they always like to build in? 5' to 3', right? So our RNA, the RNA that's going to be made, you want it to be made in the 5', 3' direction. How would you get an RNA sequence that's 5' to 3'? Well, from this complement, right? So this strand here has to be the template so that our final result is 5' to 3'. Okay? So the way that you would do this is simply take the complement of each piece of this strand. So if we were to make RNA from this DNA, we would have to go step by step. Complement of 3' is 5'. The complement of G is C. The complement of T is A, G, C, C complement is G, G, C, and then again the final complement is 3' to this. Now when you look at this RNA strand that's been created in 5' to 3', what do you notice? What does this look like? This looks like our very first sequence, right? That's CAC, CAC. And you notice the 5' and 3' orientation is the same. So this strand of DNA would be called the sense, and this strand is the antisense. So you notice that the RNA has the same sequence as the sense strand of DNA, and it is anti or complementing our antisense. Okay, so now to kind of practice this a little bit more, okay, I'll have you do some practice problems. Okay, so to practice this concept a little bit more on sense versus antisense DNA and how to get the RNA that would be created during transcription, the way that you would encounter this as a question on an exam or whatnot is you'd first know whether you're given sense or antisense DNA. Okay, and it would always be given to you in the 5' to 3' direction. So in our first bullet point, we're looking at sense DNA. And like I said before, as soon as you see sense DNA, look at that S and remember same. So when you're given a sense DNA sequence, your RNA should look identical to that. Same number, same letters everywhere, except since it's RNA now, any T should become a U. Okay, so if we're given sense DNA 5', C, A, G, T, 3', then we have to make the 5' to 3' RNA. Since this is sense DNA that we are given, our RNA should look the same. So it would be 5', C, A, G, and then because it's RNA, U instead of T. And it continues to the 3'. Okay, so notice the DNA and the RNA look the same everywhere except for when you have a T in the DNA. Now it's a U. Other than that, it's the same. The other way you may encounter this is if you see antisense instead. And remember, the moment you see antisense, anti tells you that you should be complementing everything, including the numbers. So if you are given an antisense DNA sequence that is 5', C, A, G, T, 3', then you go one by one and complement each of these, okay? So it's not going to be the same number or letter anywhere. It's the complement. So when you ask yourself, what is the 5' to 3' RNA, right? Well, don't worry about orientation yet. First, simply complement everything, including the numbers. So the first thing you would do is instead of a 5', the complement is now 3'. Then G instead of a C, the complement to A is U, C complements the G, A complements the T, and then 5' complements the 3'. Okay, so for each number or letter, you're doing the opposite, the complement. Now, keep in mind, the question will always ask for the final answer to be written in 5' to 3'. So if you give me this, then you are currently incorrect, because that is not the 5' to 3' RNA. So the last thing you always have to do is make sure to rewrite the final answer 5' to 3', simply reading this from right to left. So you'd write 5' A, C, U, G, 3', just like you see here at the bottom of the slide. So 5' A, C, U, G, 3'. Okay? So once again, anytime you're given sense DNA, you simply write the same numbers and letters exactly the way they are and replace any T's with U. If instead you're given antisense, then every single number or letter must be the complement, and your final answer must be flipped around to be 5' to 3'. Okay, so I always recommend doing the antisense in 2 steps. That way you don't accidentally miss a letter or write anything incorrectly if you try to do it in 1 step. Okay, so now I want you to practice this concept, because again, this is something that students usually get some points off during the semester for. So I'm going to give you some extra practice now.
Okay, so this concept can be a little tricky sometimes. Students get confused, or it's easy for any of us to make very simple mistakes when it comes to solving these problems. So I just want to make sure that each of you have a solid grasp of what to do when you encounter these problems. You will see them again, nudge, nudge, wink, wink. So I'm going to give you two practice problems right now. This first one is dealing with a sense strand of DNA, and the next one will deal with antisense. I want you to please send me both of these practice problems in the Remind app. And so I want you to take a picture of the slide, please, and send me the picture of the slide and your response. Again, notice that it is asking for the answer as the 5' to 3' RNA. Okay, so please send me practice problems 1 and 2 right now when you get to these slides in the Remind app. And here is the second practice problem. Again, please make sure to send me a picture of the slide with your answer. Now, when we talk about gene expression, there are two different terms that you may hear depending on whether we're talking about eukaryotes or prokaryotes. The first one is monocistronic. And when you look at the word monocistronic, you notice mono means one. So eukaryotes, in this case, you would see a monocistronic system in which you have one gene coding for one mRNA, which then codes for one protein. Prokaryotes, on the other hand, have polycistronic systems in which you end up with a cluster of genes called an operon. So in other classes and in this class, you study it a lot of times, the lac operon, the trip operon. And this cluster of genes ends up coding for a long RNA, so a polycistronic mRNA, that then codes for several different proteins. Okay, so that's why it's poly, many proteins from that single cluster of genes that's under a single regulatory region. Okay, so notice with eukaryotes, the one gene is under one regulator area, and then here you have that one cluster of genes under one regulatory region, and then here you end up with several proteins. Now, another term that ends up being talked about in gene expression is the ORF, or open reading frame. And that basically is the sequence that's going to code for the protein. Okay, so here when you see the structure gene and the structural genes, this blue area would be the ORF, the open reading frame. And then in front of that ends up being the one regulatory region, and then the sequence right after the ORF ends up being the other regulatory region. Okay, so you end up having the ORF sandwiched between regulatory regions.
So as you saw from the video, with transcription, just like you'll see with translation, you have the three terms initiation, elongation, and termination, and you have to know what's going on in each of these steps. So initiation is how things get started. So with transcription, there's two key players in prokaryotic initiation. The first one is the promoter, and next to the word promoter, I want you to write DNA sequence. So the promoter is a DNA sequence that basically tells the RNA polymerase, hey, I'm a gene that needs to be expressed, come here and bind to me. So the promoter is what gets recognized by RNA polymerase in order to be expressed. Every single gene needs a promoter in order to be expressed. Otherwise, RNA polymerase, which builds the RNA, would never be able to arrive at that gene to transcribe it. Now, the RNA polymerase that's responsible for actually transcribing or making the RNA is made up of two subunits, and there's a trick to remember which subunit's doing what. For sigma, I always write sigma sees the promoter. So I underline that S and think sigma sees the promoter, because that's the subunit that actually recognizes and binds to the promoter to start transcription. Whereas the core, that's more involved with elongation. The core constructs, okay, that's how I remember it, core constructs in order to build the rest of the RNA. Okay, so sigma sees the promoter and then the core constructs, and the core constructing is more the elongation step. So just to make sure that you have the memory trick written down in your notes, I've written it on this extra slide here. So sigma sees the promoter, and so it starts transcription. That's part of initiation. So whenever you're asked about initiation of transcription, remember, sigma subunit of the RNA polymerase and the promoter. Okay, those are the two things involved in initiation of transcription. Whereas elongation, core constructs the new RNA strand, and that is elongation. Okay, so this is just another recap of initiation and elongation with a couple of extra details. So basically what happens is sigma sees the promoter of the DNA, it binds, and the core enzyme will then open up the DNA. 6 to 9 bases will be synthesized while at the promoter, and then at that point sigma is no longer needed, and so it pops off, whereas the core continues elongating the new RNA strand. Okay? And the reason why I write RNA pol is the rock star is to once again emphasize that RNA polymerase does not have the limitations that we saw with DNA polymerase. Now, with everything in life, everything that has a start needs to have an end. So for transcription, termination occurs when the RNA polymerase hits what's called a hairpin structure. And this figure right here shows what a hairpin loop looks like. And basically what happens is the RNA polymerase gets destabilized by that hairpin loop, and it causes it to pop off. Okay, so when you think about transcription, remember initiation is the sigma subunit and the promoter. Elongation is the core subunit of RNA polymerase. And then termination is the hairpin structure that destabilizes RNA polymerase. Some differences in terms of eukaryotic transcription is that for eukaryotes, you see this long list of key players. You have multiple RNA polymerases instead of just one. Okay, you don't need to memorize the 3 exact types. Just know that eukaryotes have multiple RNA polymerases instead of just one. And the eukaryotes do not have a sigma equivalent on their RNA polymerase. I want you to circle, star, highlight that bullet point right here. Eukaryotes do not have sigma. Why is that significant? Well, in prokaryotes, what does sigma do? Sigma sees the promoter. It binds, it allows RNA polymerase to bind to the promoter and start transcription. Eukaryotes do not have that. So instead of sigma, eukaryotes need a series of transcription factors. Okay, so whenever you see transcription factors, these are in eukaryotes to help polymerase recognize and bind or initiate transcription. Okay, so transcription factors serve the purpose that sigma would have, to recognize and bind the DNA to start transcription. This is to show you the various transcription factors that exist. Okay, so there's a whole bunch of them. This big pre-initiation complex needs to form in eukaryotes. Again, it does not need to form in prokaryotes because prokaryotes have what? They have the sigma subunit of the polymerase. So prokaryotes do not need this. Eukaryotes need all of these players to help recognize and bind to the promoter. You don't have to memorize these guys. The only one I want you to know here is this guy here, TF2H. That's a transcription factor in eukaryotes, including your own cells, that will phosphorylate the tail of RNA polymerase to allow the polymerase to move for elongation. So if you visualize this big structure here is a whole bunch over here as well, a whole bunch of transcription factors. What TF2H does is if you picture this big blue blob as RNA polymerase, TF2H will phosphorylate the tail. And if you think about that, I'm going to show you on the next slide why that's important. Because if you think of phosphorylation, phosphate groups have negative charges, right? So that TF2H is putting a whole bunch of negative charges on this piece of the RNA polymerase. So by down here, you can see it. A whole bunch of phosphates is a whole bunch of negative charges on the polymerase. What do you know about DNA and the charge of DNA? Well, DNA, this DNA strand that the polymerase is going to read, is negatively charged. So if you're suddenly adding some negative charges to the polymerase, it'll allow that tail to slightly repel the DNA so that instead of being tightly bound, it'll be able to slide across the DNA and read the gene in order to express it, because you need the polymerase to move across the gene. You don't want it to just stay stuck at the promoter. Okay, so the phosphorylation of transcription factor 2H is very important for the movement of RNA polymerase, which is very important for elongation, for actually making that RNA from the DNA. Okay? So you don't have to memorize all of the other transcription factors I had on the previous slide. Just know the significance of transcription factor 2H is to phosphorylate RNA polymerase so the polymerase can move across DNA and transcribe the genes. Okay? If you're still a little unclear about that, please send me a remind message and I'll clarify it. Now, I want to just make a quick point that all of the regulation that we've seen so far is what we call upstream, meaning the regulation was occurring in front of the gene. Okay? I want you to be aware that in eukaryotes, there are also regulators after the gene. So this is our gene here. These are upstream regulators to the left, and in eukaryotes, you have downstream regulators to the right. What we call these downstream regulators are enhancers. So enhancers are kind of unique because they're far away from the gene that they're regulating. And basically what an enhancer will do is that it will bind, so picture it over here far from a gene, it'll bind transcription factors. And then DNA will loop around, which you see in the picture here. So the transcription factors bound to the enhancer, once the DNA has looped around, these transcription factors are now brought nice and close to the big transcription complex where we have, the activation of the gene. Okay, and remember that in eukaryotes, you can't have transcription without all of these transcription factors, which are not necessary in prokaryotes because, again, what do prokaryotes have? They have the sigma subunit to take care of all of this. So again, in upcoming lectures, we're going to end up having a whole lecture dedicated to gene expression regulation. But there are, still some terms that I want you to start getting comfortable with now while we're starting to introduce the topic more and more. What I want to look at here is some eukaryotic regulators. You can have positive and negative regulation. Again, this is going to be happening in the regulatory region that I mentioned you have in front of the open reading frame or the actual gene, you can call it the coding region.
Okay, so the regulatory region is directly upstream of that, and includes, you see the promoter region, the promoter proximal element. And so the types of proteins that you could have binding here for positive regulation, so increasing activation, you would have transcription factors, activators, HATs, which are histone acetyltransferases, so and chromatin remodelers. And what those guys do is they help loosen the DNA away from the histone proteins, because remember, eukaryotes have chromatin, they have nucleosomes. The DNA is tightly attached to proteins that are, blocking the gene and the promoter region. So things like HATs, histone acetyltransferases, and like chromatin remodelers, they help with positive regulation because they help loosen that DNA away from the histone proteins. Now, you also have negative regulators in eukaryotes, repressor proteins, HDACs, some things called DSIF, NELF. And so we're going to talk about those in more detail again in later sections. But basically, things like HDACs are histone deacetylases. So they're opposite of what we talked about in positive regulation. Instead, in negative regulation, deacetylases and other proteins may be tightening the grip of the DNA to histone proteins and, blocking the accessibility. The one that I mentioned, the DSIF-NELF, that's what's called DRB sensitivity inducing factor and negative elongation factor. That basically binds to the polymerase and pauses transcription at initiation until those guys get, phosphorylated. And so then you can, get rid of that repressive effect. Now, one of my favorite regulators, and I want you to circle star highlight this because I like things that are different or unique or that go against what you usually think of for, some kind of topics. And so enhancers go against what we usually think of with regulatory proteins. Because so far, all I keep saying is, oh, the regulators, they bind upstream, right in front of that gene start. Enhancers are different. Enhancers, first of all, these are not proteins. So the what we've been mentioning over here, positive and negative regulatory proteins, enhancers, the first thing I want you to write next to it is enhancers are not proteins. Enhancers are actually DNA sequences. And when you look at the location of the gene, the regulatory region that we've been talking about, like the promoter, is right in front of it. The enhancer DNA sequence is far downstream from that gene. It's far away from the gene that it's regulating. That's unique and distinctive. And so you'd be like, wait, why is it all the way over there if it's supposed to help this gene right here? Well, what it does is it binds. It binds several transcription factors, and then the DNA loops. So this enhancer that was all the way over here, when the DNA loops, look, now it's close to the promoter region. So it's bringing transcription factors to the promoter region. Okay, and we're going to see this more in detail when we go over the mechanisms a little bit more in the upcoming lectures. But I want you to really remember enhancers because I think it's kind of cool that they go against what you would normally think, logically of regulation. You would think, oh, the regulator's right next to the gene. Not always. Oh, you might think the regulator should be a protein. Not always. Enhancers are DNA sequences and they are far away from the gene. Okay, so we talked, a lot of in terms of making that RNA, and now I want to point out, after we touched upon a little bit of regulation too, I want to point out that once you have that RNA made, it still needs processing to be functional. Basically, when you finish with transcription, that's not the end of the story. And so now you need to proceed with that RNA to make it fully functional and stable. Because we like things that are fully functional and stable, unlike us sometimes. Now, when we talk about eukaryotic RNA and, helping it become that mature RNA, because we all want to be mature one day. I'm still waiting. Anyway, when we talk about eukaryotic RNA, to make it mature, there are three steps that I want you to be aware of. First, a 5' cap has to be added. Then on the other side, a 3' poly A tail. And finally, you need splicing to occur. Now, when we look at each of these, I want you to keep in mind, two questions for yourself as we go through the next few slides. What is being done and why? What's the function? Why is it important to add a 5' cap and a 3' poly A tail and have splicing occur? Okay?
So again, with eukaryotes, I want you to remember that they have to process their RNA, that this requires 3 different steps. The first step is that eukaryotes will add what's called a 5' cap, meaning that on the very beginning of the RNA for a eukaryote, they will add this little reverse orientation GTP. Okay, this is important for stability. Okay, so this functions as stability because by putting this GTP that you can see here in an upside down orientation, it basically gives the appearance that the 5' end of this RNA is actually a 3' end. The reason why that's important is because in the cell of eukaryotes, there are a lot of 5' exonucleases, meaning that there are a lot of enzymes that will go around and chop up 5' ends of RNA. So by putting a 5' cap, that makes the 5' end look different, these enzymes can't recognize the RNA and won't chop it up. Okay? It also helps with the movement of this RNA for things like nuclear transport, but don't worry about that for now. Just know the 5' cap gets added on eukaryotic RNA to protect the RNA from nucleases that would chop it up. Okay, so the second step in eukaryotic RNA processing is for the eukaryotic cell to add what's called a poly A tail. So what this means is that on the end of every RNA, the eukaryote will add this very long string of A's. Okay? The reason why it puts this long string of A's is kind of like what we mentioned a minute ago. It's for increased stability of that RNA. It basically helps prevent degradation of the coding region of the RNA. Because think about it, as this RNA is moving around through a eukaryotic cell, like I said, there are going to be a whole lot of enzymes that will try to chop up the RNA, and they usually do it from the ends of the RNA. So by having this long string of A's at the end of the RNA, these enzymes can chop, 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 chop without actually chopping any part of the coding region, any part of the RNA that actually codes for proteins. Okay, so you don't have to know the details of how this is formed, okay, all these bullet points on how it's formed. Just know that eukaryotes have to add a poly A tail, and then it's to protect the coding genes, the coding part of the RNA from getting chopped up. The last processing event to mention is the idea of splicing. And the reason why splicing occurs in eukaryotes but not prokaryotes is that eukaryotes have introns and exons, whereas prokaryotes just have their exons, which are the coding areas. Okay, so in RNA, when you hear exons, those are coding regions. Introns are non-coding. So what splicing basically is that a spliceosome, a special structure, basically needs to cut out any introns and then glue together the exons. Okay, so spliceosomes will cut out any introns and glue together the coding exons. Okay, and to demonstrate what that looks like, we have a little video.
The following animation will describe the process of RNA splicing, an important step in creating the mRNA that is involved in protein synthesis via the process of translation. Key factors in this process include RNA, possessing introns and exons, and the spliceosome. Here we see an RNA molecule with a single intron. Several signals exist within the intron that are used in the splicing process. From the 5' end of the intron, these are GU, the A branch site, a pyrimidine-rich region, and the 3' AG. The GU and AG sequences define the beginning and end of the intron. Splicing is mediated by the spliceosome, which consists of several protein RNA complexes. The first step involves two complexes that bind near the GU sequence. The RNA is then looped, and 3 other protein RNA complexes bind. This final complex then undergoes a conformational change. Introns are non-coding RNA sequences that must be removed before translation. The process of removing the intron is called splicing. The intron is then cleaved at the 5' GU sequence and forms a lariat at the A branch site. The 3' end of the intron is next cleaved at the AG sequence, and the 2 exons are ligated together. As the spliced mRNA is released from the spliceosome, the intron debranches and is then degraded.
Okay, so the last topic that I want to talk about today is RNA-mediated gene silencing. And the purpose of this is basically to regulate cellular processes. And, that can include things like apoptosis, and cell proliferation, even development and differentiation of cells. And ultimately, when you have RNA-mediated gene silencing, it can even serve as oncogenes and tumor suppressors. And what's pretty cool about this concept is that if you think about it, if it's RNA-mediated gene silencing, it can even help when you think of it being a defense against RNA genome pathogens. So, for instance, viruses that have an RNA genome. Now, when we talk about RNA-mediated gene silencing, the 2 examples that I want you to know are siRNA and miRNA. So that's short interference RNA and microRNAs. And so the difference between them, siRNA, I want you to think Si as in slicing, because siRNA will cleave mRNA to inhibit translation, because think about it, if you chop up the mRNA, it cannot be read, okay? So you can't get proteins made. The other one, miRNA, this one will physically stick to the complementary mRNA. And so you can think of it as the miRNA will base pair complement to the mRNA. And so if you have the microRNA wrapping itself and gluing itself to the mRNA, well, now the cell cannot read that mRNA. And so you can think of that as a form of inhibiting gene expression by post-transcriptional regulation. Okay, so make sure you know siRNA and miRNA and the difference between how they're working.
And that is it for today. I know that was a lot of information. Please contact me as always if you have any questions or concerns. Please make sure to get those practice answers into me. And please, again, if you can always send a picture of the slide that you are either answering a question about or that you have a question about, it helps me to better answer and to help you navigate the problem. All right, that's it for today. Thank you and have a great day.